Welcome back to Bio180 lecture series. My name is Dr. Christensen, and today we are going to be continuing our exploration of cell processes. This is our 5th unit in Bio180. And so we've already gone over various aspects of membrane transport, and now we're in the middle of our section on cell communication. We've talked about different types of cell signals, so today we'll be focusing on receptors in preparation to talk about transduction pathways, and then we'll move on to our final material talking about cell division. But today we'll focus on receptors. And so we want to talk a little bit about the receptor-ligand interactions. We'll talk a little bit about receptors and their KD, or dissociation constant, and then we'll go through mechanisms for different receptor types that are quite common in cell signaling. So let's get started with just a few notes about receptor-ligand interactions. So, receptor-ligand. If we have a membrane, and so there's going to be some kind of receptor, so we'll label this as our receptor. And then the receptor is going to have some kind of ligand that binds with and associates. And so that ligand is going to come into that binding pocket, and so we'll call this it's called the signal binding pocket. So that ligand is going to come in and bind in that pocket. And there's a couple notes that we want to have in there, is that the ligand and receptor interaction. Receptor ligand interaction is based on weak bond formation. And so that ligand is going to come in here, and it's going to be held in that binding pocket with weak bonds. And then depending upon the number and the strength of those weak bonds, it can not only come in, but it can also come off. And so ligand binding is dynamic. Because it's based on weak bonds, all that dynamic means is that the ligand can come in and be bound, and it can come back off. So ligand binding is rarely permanent. That said, well, let's talk about another feature, and that is ligand binding is specific. And so when that ligand comes in, there can be tens to dozens to scores of different receptors on the surface of the cell. But this ligand, that signal, when it comes in, is only going to bind to its appropriate receptor. And that's because the ligand binding is specific. So that receptor is designed to recognize that ligand based upon its shape and polarity. Remember that we're talking about the number of weak bonds that's going to hold it there, and those weak bonds have to be in the right position, so that shape has to be complementary, like a hand inside a glove or a key inside of a lock. And then it not only has to be the right shape, but you have to have the right polarity in the right positions. So for example, if we were to come over here and look back, if I had a positive ionic charge right there on the receptor, I would need to have a complementary charge right there on my ligand so that those two could bind together. If I had a hydrogen donor right there on that end, I would have to have a hydrogen acceptor in the pocket. If I didn't, if I had another donor, then that interaction wouldn't work. And so it has to be complementary both in terms of shape and polarity. There's another feature then, and that is affinity. So ligand affinity is how strongly, is a measurement, the affinity is how strongly or tightly, how tight ligand binding is. So ligand affinity is a measure of ligand binding, and it's based on the number and strength of the weak bonds between the ligand and the receptor. And we use a measurement called the KD. So measured by capital KD. And so this is called the dissociation constant. And so let's just talk briefly about that, of what a dissociation constant is. We can consider a ligand binding, or so we would say right here, we're going to take a receptor, and then I'm going to put a colon here, and that's going to be my ligand. And this is going to be the bound complex. And then we could say that this is a type of chemical reaction in which we are going to have receptor plus ligand. So these are the unbound or the free receptor, and the unbound or the free ligand. And so this is a reversible reaction. They can form the complex, or they can form these unbound or free elements that are independent of each other. Now, because we're going from the complex to the dissociated or the separated components, this is referred to in the forward direction as a dissociation. And so that would be a dissociation reaction. The complex is together and we're separating them. So if we consider this to be a chemical reaction, then we've also learned that there's some kind of equilibrium constant, K, for that reaction. And because this is, so we call this a KEQ generally, an equilibrium constant, but we have a specific type of reaction, which is a dissociation reaction. And so we're actually then going to call this the KD, the D standing for dissociation. Now, the KD of this reaction then, as we learned earlier, is equal to the concentration of the products over the concentration of the reactants. And so we would write that up here as the concentration of receptor over the concentration, or the product of those two, over the concentration of the bound complex together. And that would be the KD. So this then represents, to remind us a little bit about equilibriums, this KD then gives us the constant ratio of products and reactants at equilibrium. Okay, so we just want to remind you a little bit about this, and this becomes significant, and we want to get back to understanding how tightly a ligand might bind to a receptor and what that means, and what does the KD show us. So suppose, for example, we had a receptor that had some kind of KD, and let's just say it was equal to 10. All right? So if my KD was equal to 10, that would mean that at equilibrium, I would have, if remember that's the concentration of reactants and ligand over my. So what I could write here is that I would have receptors and ligands. if I put a 1 down there, I didn't change the number, I just gave it 1 as a denominator. The 1 correlates to my complex, and then the 10 is the product, so x times y would somehow equal 10. But no matter how you slice it, I've got more reactants and products here at equilibrium. So a Kd of 10 is one that favors the unbound form. And so this is a generalization that we can make, is that a Kd greater than 1, Kd is greater than 1 equals favors the unbound receptor. On the other hand, if we had a Kd, so let's over here consider a Kd that was equal to, I don't know, 1/100th, or we'd say that could also be 0.01. In this scenario, again, if we write up our formula, we'd have the concentration of the products, R and L, over the concentration of the complex. And so now in this form, notice that we've got, if we just look straight across at these, for every 100 reactant and ligand, or receptor and ligand complex, we would only have about 1 representation of a receptor and ligand. And so in this case, notice that we're favoring the complex itself. Now, why at equilibrium is it favoring the complex? Because at equilibrium, this particular ligand-receptor interaction must have quite a few bonds that are holding it together. So it's more likely at equilibrium to be found in the bound form than it is to be found in the unbound form. So the tighter the binding, let's make up another rule. The lower the Kd, that is equal to tighter binding. Okay? And so those are some rules that we want to know for being able to evaluate Kd. So any kind of receptor interaction, we are going to be interested in this value called the Kd for that reaction or that interaction. And then once we know that Kd, then we're going to look at it and say, well, if it is greater than 1, it favors the unbound or free form. If it is less than 1, then it favors the bound form, so the bound receptor. And we can further say that the lower the Kd number is, the tighter the binding, and then the converse of that is the higher the Kd equals looser binding. Now, you might be thinking, or a lot of students think, well, that means that I always want to have a lower Kd and tighter binding, because tighter binding must be a good thing. That's not always the case. There are cases where we actually want inside the cell a ligand that binds more loosely to the receptor. Where might you want that? Well, for example, you can look at the synapse between the nerves and the muscles. So that synapse is you have a signal molecule called acetylcholine. And so acetylcholine will cross that neural synapse and bind to the receptor. And that's what initiates the muscle contraction. So that's the signal that's used to initiate muscle contraction. However, if it were to bind onto that receptor for a long time, you wouldn't be able to have very much control over that. It would bind, that muscle would go into contraction, and it would stay in contraction until that ligand was able to release and stop the signal going forward. So if it binds on there tightly, I'm going to remain in contraction and not be able to control it very well. So what you'd really like to have is a ligand that binds to it, initiates contraction, and then comes off so that you can stop the contraction whenever you want. And then if you want a prolonged contraction, then you would just flood it with a lot more signal and keep that receptor. As one comes off, another signal molecule would go on. And so that's the way that you have a durable signal, but also still have control over it. So, take-home message is that there are some cases where looser binding is actually preferable in the cell to tighter binding. So, all this tells us is whether it's looser or tighter. It doesn't tell us whether it's good or bad. So, with that introduction to some of our ligand and receptor binding, now we're ready to start looking at some of the different receptor types that we're going to encounter in our class. So now, with that introduction to receptors in general, their interactions, and a little bit of an introduction to the dissociation constants, we're ready to talk about our different receptor types. And specifically, we are going to talk about the mechanisms of these receptors. And so, there's a couple things I want to go through with you guys first before we start these mechanisms. First of all, realize that each one of these mechanisms are processes. And so we're going to be talking about these, and we're going to be making some illustrations of these processes. And so be aware that we're trying to illustrate dynamic processes, things that are changing in a still medium on a whiteboard. And so to do that, I'm going to be using a lot of arrows, and particularly numbered arrows, to show different things happening. And so this is commonly done in the biological literature, and so we're trying to get you used to that a little bit. So we'll be talking about trying to do these illustrations and use arrows. And I might draw a protein, and then if I draw another protein right next to it, that may be the same protein, just in a different form or at a later time. So I'll try and point that out as we come through. As you go through and study these, we will show you how to do some illustrations. And then you'll also want to learn sort of step-by-step, how to write them out step-by-step of what's happening. The other thing I want you to note in these processes is we will talk first about activation of the processes, but realize that just as important in cell signaling processes is the deactivation, and how you turn the signal off. And so with that little bit of primer then, we'll get started on our first mechanism, which is going to be ligand-gated ion channels. So, we're going to draw a membrane here. And then in that membrane, we are going to start out with an ion channel. Okay, so here's my representative ion channel. And so here's the channel itself. So the idea is that we'll have some kind of ion, like, let's take potassium ion. And the idea is that it would come through here. But we're going to start with this channel. I guess we should start with it this way. We're going to start with the channel in its closed or inactive form. So we're going to say that the channel is closed or inactive. Here's my potassium ion. And then the other thing that we need here, so the first thing that's going to happen is we're going to have some kind of ligand. And that ligand is going to enter into this ligand binding site. So that's going to be step 1. We'll put a number 1 right there by that arrow. And so the first thing that happens is that the ligand is going to enter the ligand binding site. Now, a lot of times what's happening with these mechanisms is it's cause and effect. And so you want to be watching for the cause and effect. So we have some kind of cause, the ligand is going to bind. What's the effect of that? Well, in this case, what happens is we're going to draw this arrow, and that's going to be number 2, is we get a conformational shift in our protein. And so let's try and draw what that looks like. Okay, so what's happened in number 2 is we've gone through a conformational shift. And really what's happened is that this, the way I drew it, this reminds me of this, the way I happen to draw this one, it reminds me of those German beer tankards that have kind of the lid on them and then the little thumb kind of tab on the back there. And so what's happening here is that lid is being controlled by this tab, and when the ligand binds, it attracts that back end of the lid down, and that causes the whole lid to go from closed to open. And so now we have an open channel, and that's going to allow step 3, which is with the open channel, our potassium ion is able to go through the channel. So that would be step 3 with that arrow. Now, our potassium is now going to enter in the cell, so that's successful activation, and we've kind of reached that point of activation. So now we come back and say, okay, if we've activated it, now what would the 4th step be? How do we deactivate it? And for the answer, the first thing we're going to do is remove, so in step 4, that ligand, remember, is dynamic. It binds, but it also unbinds, depending upon the number and type of weak bonds it forms. So programmed into its structure is kind of a half-life, or how long that ligand on average will bind and keep that channel open. So just on its own, by being hit by other things, that ligand is going to leave. And then when that ligand leaves, it's the thing that's causing that gate to be open. And so step 4 leads to step 5, which is a return to the inactive conformation. So this is the active or open conformation, and then this would be the inactive or closed. Conformation. And so we can see this particular mechanism has 5 steps. The ligand binds, that causes a conformational shift to the active or open conformation. Potassium in the open conformation is able to travel through that pore. Eventually that ligand will leave, and that will cause it to return to the inactive or closed. So that's the illustration with my arrows. We've gone through both the activation and the deactivation. What does this look like in a step-by-step translation? So to do the step-by-step translation, I'm just going to write what each one of those arrows does. So in step 1, we just have ligand associates or binds with the channel ligand binding site. And then in step 2, what do we say happened? Well, that ligand binding caused a conformational shift. So we have a conformation shift. And we've already talked about this word earlier. That's where the protein is actually going to change shapes. from closed to open. And then in step 3, we are going to have the selected ion, in this case, potassium, travels. We learned in our last unit that this is a passive transporter, so it's going to travel down the gradient. And in this case, I guess really what we'd say is when that potassium enters, in this case, we're going to have a signaling event. We don't have much going on right here, but this would lead to a change in membrane potential. So it leads to some kind of effect. And that's different for every mechanism. And this might be what leads to the next signal in the process. But for us, step 4 is going to be the ligand releases. From receptor. And then the receptor returns to the closed conformation. All right, and so that's our explanation of how a channel is going to work, or a ligand-gated ion channel. So that's one of our receptors. For the next receptor, we'll come over here to the board, and we will draw another membrane. So this is my plasma membrane. And then for this particular receptor mechanism, we also have to draw a nucleus. So remember, nuclei actually have a double membrane. So there's one membrane, here's the other one. We've got a nuclear pore represented in there. Okay, so here is my nucleus. That's the nuclear envelope, and I've got a nuclear pore right here in the middle. Now, in this case, I'm still going to have some kind of receptor. Now, this mechanism is called intracellular. Intracellular receptors mechanism. And what's unique about intracellular receptors is that they are not actually located in the membrane. Okay, so here's my intracellular receptor. That's one of the players. And then we're going to need a ligand. Now in this case, the ligand itself, we're going to put outside the cell, and that's going to be our ligand. And in this case, the ligand itself has to be somewhat hydrophobic. So these are And the reason they have to be hydrophobic is because this ligand has to penetrate that membrane all by itself. So it has to penetrate that plasma membrane and then come inside, and again, it's going to bind. So that's still going to be our first action, is that ligand, that hydrophobic ligand, is going to come in through the membrane. It's going to diffuse through the membrane and then associate with this intracellular receptor. And when it associates with the intracellular receptor, then we get a conformational shift. And so here we're going to show the bound form. So notice when the ligand binds, then this little appendage is going to stick out. And so this appendage right here, so we get a conformational shift. And then that appendage, this happens to be called a nuclear localization signal. Nuclear localization signal, or NLS for short. And that signal, now that it's exposed, allows this protein complex to come, or this receptor, to come to the nuclear pore, and it gives it kind of a code that causes the pore to open. And so here we have step 3, that the pore opens, and my protein is able to enter into the nucleus, the receptor. Now, once inside the nucleus, we have our DNA. So we'll label that as our DNA. And that bound receptor is going to associate with the DNA. Oops. So right here in step 4, the receptor is going to bind to the DNA, and then once that receptor is bound to the DNA, in step 5, we're going to get transcription. And so we're going to transcribe. This acts as a recruiter or a transcription factor to recruit the polymerase machinery and cause transcription. So we've got step 3 was entrance. So we entered the nucleus. Step 4 was bind DNA. Step 5 is transcription. Step 2, remember, was our conformational shift. And step 1 was signal binding. And so this is the response that we're actually looking for. This is the activation of it, transcription. So now our signal is fully activated, and then we deactivate it again by losing our, so in step 6, our ligand comes off. And that causes then the protein to fall off, to come out of the nucleus. And return. So several things go. It undergoes a conformational shift, and then also causes it to fall off the DNA and exit the nucleus. So that's step 6. Quite a few things going on right there. And that would be the pathway for intracellular receptors. Now, this pathway is unique in a couple ways from our other receptor pathways. One is, again, the receptor itself is intracellular. It's just inside the cell. It's not located on any membrane. That's unusual. The other feature is that in our last lesson, we learned about the 3 steps of cell signaling. You had to have reception, then transduction, and then response. And so in this case, this one receptor is doing everything. It's receiving the ligand. It's kind of transducing itself by traveling through the nuclear pore into the nucleus. And then it also creates one of our responses, which is gene transcription or gene activation. So we've got both reception, transcription, and response all happening in the same step. All the other receptors just do reception. So that's our second one. I'm not going to go through the steps. We've kind of written them down loosely on here that we did in our first one. But I'll let you in the worksheet go back and translate and write down some of the steps. And you want to be able to know both the steps, the cause and effect, and then be able to illustrate those effects and move both between the written word and the picture diagrams so that you truly understand these pathways and what happens with them. So now we have finished our ligand-gated ion channels. We've talked about intracellular receptors. We now have the 2 more complex mechanisms that we want to go over. So starting with the tyrosine kinase. So tyrosine kinase receptors. First, just a little explanation on the word itself. So kinase, if you remember, we've talked about this a little bit. Kinases are enzymes. So that's the name of an enzyme. And they transfer phosphates from ATP. And then in this case, tyrosine, that's going to be what we're going to transfer the phosphates to. So tyrosine is a specific amino acid that we find on the sequences of proteins. And so we're going to draw a membrane here. So here's my membrane. And then I'm going to have a receptor. And here's the different parts. Up here I have my signal binding domain. A domain is just a part or a region of the protein. Up here, down here I have a single 1-alpha helix. And so that's called the transmembrane domain because it crosses through the membrane. And then this circle right here, I'll put a K on it, that represents my kinase domain. So these proteins have an area on them that has this kinase functionality. And then this last part right here, that's my tyrosine domain. And so this is the letter for tyrosine, the letter that represents tyrosine in the amino acid alphabet is Y. So I'm just going to put some bonds off here with the letter Y. And so that represents the tyrosines that are on that protein. And so this would be one tyrosine kinase receptor. So what happens in this mechanism is that we are going to have a ligand, again, as in all of them. This one's a little bit unique in that most often, or more often than not, we have what's called a dimeric ligand. Dimeric just simply means that it has 2 of the same part on it. And that's actually going to become important for our mechanism because it turns out that to activate this receptor, it can't be alone. So we actually have to have another receptor. Here's okay. So here's a second receptor. And here's the way it works. This one has a kinase receptor or a kinase domain, and it also has the tyrosines. But for whatever reason, this kinase cannot transfer phosphates to its own tyrosines. It's sterically hindered, it's just not able to do it. And so it doesn't activate itself. However, if we can get that receptor to hook up with another receptor, this kinase is able to phosphorylate those tyrosines, and that kinase can phosphorylate those tyrosines. And so that's what we want to happen. And so the dimeric ligand is going to help us with that because it binds first to one of those signal binding sites, and then it will bind to and grab onto the second ligand. And then that dimeric ligand sort of grabs one, and then it moves around until it can grab another one close by it. And then because it's holding on to both of them, those two receptors are brought into close proximity to each other so that it looks like, I guess in step 3, we get something that looks like this. And so here is that dimeric ligand holding those two receptors together. Okay? So now, in this format right here, here's my tyrosines. In the third step, this kinase domain is going to take ATP. So in step 3, we're going to take ATP and we're going to make ADP out of it. And then we're going to transfer that phosphate to the tyrosine so that we get tyrosine phosphate. And that's done on both sides. So that's step 4. So we're first going to, this is in step 3, we call this dimerization or aggregation of that receptor. And then in step 4, this is tyrosine phosphorylation. Tyrosine phosphorylation. And so now our receptors are going to look like this. They're going to have their tyrosines on them, and I'm going to draw the tyrosines on the outside now just because there's a little bit more room. They now have this phosphate on it, so they're now phosphorylated tyrosines. And we will put another one over here. And that's kind of the end of the activation of this pathway. Once we have activated that receptor is now considered activated when it has the phosphates on the tyrosines. So now we have an active receptor. Later on, we'll see that another protein can recognize this phosphorylated tyrosine, bind to it, and pass along the signal. But that's going to be the end of our protein activation. We now have these receptors activated by the phosphates on the tyrosines. And that was coming because these kinase domains could what we call cross-phosphorylate or auto-phosphorylate each other. So that's the activation pathway. Now, how do we deactivate this particular pathway? Well, for one thing, we can get ligand dissociation. So in step 5, we can have ligand dissociation. Now, in this case, just dissociating the ligand isn't going to do anything. These now will split apart, but each one has still been activated. So while that's a start of deactivation, it will keep us from reactivating. We're not done yet. To deactivate this, we have to remove that phosphate. And so in step 6, we are going to have another enzyme called phosphatase. And phosphatase is going to come up and it's going to cleave off that phosphate. So we'll call that step 6. And phosphatate is going to cleave. Phosphatase is going to cleave the phosphate off of here, and then we will just have a normal tyrosine kinase. We remove the ligand right here, so these can now separate, and our signal is fully deactivated. Okay, so that's how a tyrosine kinase receptor works. Our last mechanism is actually the most complex one. And so this is going to be a G protein coupled receptor. So if we draw our membrane, the receptor in this case is called a serpentine receptor or a G protein coupled receptor. And so it has a ligand binding domain. Okay, so it looks something like this. Now the key here has a couple of different domains like our tyrosine kinase. So we have the ligand binding domain. And then right here we have our transmembrane domains, but notice there are 7 of them now. 1, 2, 3, 4, 5, 6, 7. And that is a characteristic of all of these G protein coupled receptors. They have 7 transmembrane domains. They kind of weave back and forth across the membrane, and so they're sometimes called serpentine receptors or snake-like receptors for that reason. But it has 7 transmembrane domains. And then down here, I specifically drew this kind of loop in it, and that is called the G-loop domain. And again, when I write the word domain, that is just a specific part of the protein that has a unique function or shape to it. And so this is what we start with. Another player that we have in this particular pathway is called the G protein. So the G protein the protein looks like this. It is a lipid anchored protein. So it's got a lipid anchor. It's attached to the membrane through this lipid anchor. And so that's the lipid anchor on it right there. And it has 2 parts to it. One of these parts is called the alpha subunit. And it actually has 3 parts to it. And the other 2 parts are called the beta-gamma subunit. And the beta-gamma subunit are often just called beta-gamma. They're spoken together because these 2, once they associate with each other, rarely separate. So they're pretty tightly bound to each other. And then we also have the alpha subunit has a binding pocket for a molecule called GDP. Now, this is GDP, this is guanosine diphosphate, so we've seen this when we learned about nucleotides. And in its normal inactive state, the alpha subunit is bound to GDP. With that introduction, we have two players in this pathway then. We have the G protein coupled receptor and then the G protein itself. So let's write that down here. This is the G protein. This is the receptor. Notice that the receptor then is going to be coupled or paired with the G protein. So the receptor, that's how it's got its name. It's a G protein coupled or paired receptor. The G protein itself is named because it binds onto guanine. So that's why it's called a G protein. That might be helpful to remember that this pathway uses GDP, and then remember our tyrosine kinase receptor actually used ATP in it. So now that we've got those two players straightened out, let's go over our pathway. The first thing that happens, again, is we're going to have some kind of ligand bind to the receptor itself. In this case, the receptor then undergoes a conformational shift. So I'm going to draw an arrow way over here and redraw my receptor. Okay. So notice what's happened is this G loop, when the receptor bound, the conformation shift caused the G loop to become exposed. So this would be step 2, and we have a conformational shift. And that exposes the G loop. Now, once that G loop gets exposed, that acts as a binding site now for my alpha subunit. And so here is my alpha subunit and my beta gamma. And we have GDP bound in it. And so now it can fit into this. Before, this loop wasn't down, it was tucked up and it couldn't fit. But now we can, in step 3, get association. So now my G protein itself is going to associate with my G protein coupled receptor. And so now we get something that looks like this. So I better draw a ligand in here. Now, so it now looks like this. Now what happens in this next step is that the GDP that was in here, in step 4, this G loop causes a slight conformational shift in the alpha protein, and so the GDP actually exits. So that is step 4. And in its place, we are going to get a new molecule of GTP. So this is triphosphate, and the triphosphate is going to come in and fill that void right there. So this would be step 5. The GDP exits, it's pushed out by that G loop interaction, and that leaves an open position for the GTP to come in and fill that site. When that happens, We'll draw our membrane again. And I'm just going to draw the alpha protein. So now when GTP fills that active site of the alpha protein, it undergoes a conformational shift. Conformational shift. And so it changes shape. And one of the main things that it does when it changes shape is it deforms so that the beta gamma subunits that were previously bound to it can no longer bind. And so these become free, and they can kind of go where they want to. The alpha can kind of go where it wants to, and so you get separation. So this would be step 7. You get separation of those two components. Now, separation of these components means that they'll travel in the membrane, and the alpha by itself, or in some cases the beta gamma by itself, can then bind to some other thing. So in step 8, we get binding to the next step. Okay? And that's kind of going to be the ultimate activation. So the ultimate activation of this signal is an alpha, is a dissociated or a separated alpha subunit from a beta gamma subunit. That's the whole purpose of what we're trying to get to in this receptor pathway. So that's the activation. Now, how do we inactivate it or deactivate it? Well, to deactivate it, it's going to depend a lot on this alpha subunit right here. And so this alpha subunit has what we call GTPase activity. And what GTPase means is that it has GTP hydrolysis ability. So the alpha subunit can actually hydrolyze this GTP into GDP. And it can do that all by itself. It has that inherent activity, but it's not very good at it. So it takes it a while to do it. So the GTP usually sits on here for, I don't know, let's say a minute or two. It's probably faster than that in terms of the cellular timing of it. But it sits on here for a while, and then eventually it gets hydrolyzed by the alpha subunit itself. So this is kind of like, I'm going to put a watch here and some watch hands. There's kind of like a natural timer on this subunit. It will hold the GTP for so long, and then it will cleave it, and the phosphate comes off. So we'll get the phosphate coming off, and then that creates or turns it into GDP. Well, when GDP is bound, it comes right back down to that version. So now we can get reassociation of the alpha and the beta gamma subunits once it hydrolyzes. So hydrolysis leads to reassociation. Then the other thing that has to happen, it will just come back and re-exchange. So we'll call this GTP exchange. As long as we have a ligand and a G loop, it will just cause a re-exchange. So the other thing that we have to do is we have to remove the ligand from the binding site. If we remove the ligand, we'll tuck that G loop in, and then the reassociated G protein will not be able to carry out further reactivation of it. So I know there's a lot more steps. So remember, I said this was the more complicated of these pathways. But that is a G protein coupled receptor pathway. This particular type of receptor is actually really important inside the human body. So, for example, it's this type of receptor that's associated with a lot of our senses. For example, sight, our ability to see things, our vision, is controlled by a member of the G-protein-coupled receptor family called rhodopsin. And our smells, our olfaction receptors, the things that receive scent, are also G-protein-coupled receptors. Some of our taste buds are G-protein-coupled receptors, as well as some pain receptors and other things. So this is a really large and important family of receptors that you'll see over and over again in your study of biology. So understanding its activation and deactivation is pretty significant. So to wrap up, we've now finished talking about tyrosine kinase receptors and G-protein-coupled receptors. And that's going to conclude our video presentation today on receptors. We'll see you next time when we talk about transduction pathways. We'll take these receptors and show you the next steps that follow those. Until then, we'll see you.